And it's quite ideal that the fossil sampling matches also the intended sample regime, for example, diversified or random, that we are going to be a bit more about that this afternoon. So first, it's important to study our fossils and then select the characters to score. Why is this important? Because the fossils are discovered all the time. And so it may be new characters that we would like to score and not using only previous data sets of fossils that have been found for our group. So more fossil data means more paleontology background that we need to know. And also this geology that states about the ages of the family. We don't have to forget about that. The times of the different straits are also changing over time according to the new techniques that are used. So what do we do? We can do some basic search of the fossils that are available for our group. And for example, we can use the paleobiology database for that. This is an example that if I search there by different categories, for example, according to country, or in this case, according to the taxon name, we can then see the different records that appear there according to different bibliography, different authors. But it's always very important to check each of these references in particular, because many of them, sometimes they are wrong, like they are, for example, pointing to some particular age or fossil actually not from our family. So we have to check beyond these works and also analyze the repositories. Why is this important? Particularly important because, for example, we can see the description of a couple of fossils from Russia, say for example, and then when we go there to analyze those fossils, we can find a huge amount, much more fossils that are interesting and could be assigned to our group of study. So it's really important to analyze and to search the museums because there are much more fossils there that we expect usually. So let's see a little bit about the nature of the fossil record. As I said before, it's not complete, sometimes because of preservation reasons, but also it's about how are collected, how are the explorations focused in some parts of the world, as well as the funds are available for more explorations, for example, in North America or in Europe, in other continents. But first, let's see some of the conditions for a plant to be fossilized and preserved. And some of them is, for example, the locality. That's pretty obvious, that the region where the deposition is taking place. But also sometimes if this plant has been transported to a body of water, it could be more fragmentary, the nature of that fossil. And the water, the presence of this kind of body of water is another condition to allow the preservation of several fossils, of plant fossils. And we're going to see a little bit about the different kinds of preservations. So all this is related to taphonomy. And the taphonomy is this study of the process of preservation and how they affect the information that is available on the fossil record. So according to the preservation, it's also what characters we are going to be able to score. According to preservation, we'll see if some characters are going to be biased, but that is preserved, or it could be in a very unaltered state that we can score several more characters, or even have some details about the cell that are contained in these fossils. So about taphonomy, so if you know, as I said, how your study of group fossilizes, you will see that you can predict what are the morphological traits that you could score. This is quite useful for comparative anatomy and morphology, and also to evaluate the different hypotheses of homology, and at the end also building the character matrices. For example, you can see here two different fossils that one is one seed and this is a fruit. The difference beyond the structures that you can see is that this is in an altered state. It has volume, for example, and also in this one is a compression. So it's entirely compressed. So if we compare sometimes characteristics of these fossils, and especially, for example, if we find a seed inside of this fruit, do you think that we can actually compare how is if the volume of these different fossils is included in the fruit with a in an altered state? What do you think? Something that we can codify? Or it will be biased? What do you think? If this is a compression, so the seeds contained in this fruit, if we can find some, are going to be compressed because of the preservation, actually, and not because of the nature of the seed. But in this case, we can see because it's in an altered state, if it's compressed, we have some volume, we can do CT scans, we can do another type of analysis. So let's see a little about kinds of preservation. First, we have compressions, the most common for flowers, membranous structures, leaves. In this case, the plant part is buried and is covered by sediments of various kinds. So the result is actually a flattened structure, sometimes carbonaceous as well, with the outlines and surface features of the plant part. Usually there is no much internal cellular detail that is preserved to be scored there. So as I said, this is the most common preservation for leaves and flowers, sometimes for fruits as well. And usually the sediments are covering these structures. It's shale, sandstone, it could be diatomous earth. And when this matrix is with finer grains, that means that it's a better preservation. Okay. Something that is also very useful is that sometimes in these compressions, we can also see cuticles. And these cuticles, it's these waxy layers that covers like all the leaves and other organs, all the plant actually, except the empty roots. And we can see in this way some of the stomata structure, the occlusive cells, as well as the epidermal cells that could be scored, as well as frequency, shape, and the distribution of hairs, for example, that could be useful also to score and describe fossils. Something interesting is that there is this database that is for cuticles of taxon fossils, and you can see there a huge amount of different cuticles you can compare. Something important about the compression and here is we have organic material, of course. Then we have the coal. Like it's a kind of compression, but there is an extensive accumulation of the plant material in this base of water. It's gradually compressed, so it's turning entirely into coal. And in this case, there is no internal cell details. And then we have an impression. The impression type is actually like a compression without organic material, because basically it's a negative of a compression. So the sediments that cover and compress the plant they harden, and when they split open where it was the fossil, they show this negative imprint of the plant part. As I said, it's kind of a compression without organic material, so there is no cellular detail. Other type of preservation will be a three-dimensional mold. And in this case, the sedimentary material surrounding a plant part can harden to form it when it was only a little of compression. So what happens is then the plant part, the organic material, disintegrates, and it's filled with other sediments, forming this case of cast. So as a or there is no organic material, no cellular preservation as well. This is very common to trilobites and other also animals, for example, not so common in plants. And then we have the petrification. This is common in bones, in woods, and in this case, the organic material of the fossil is totally replaced by minerals, but there is a very good preservation when it's like the shape and how was the cellular in there. And this process involves a combination of actually two processes. So the first one is the permineralization. That is the groundwater dissolving the material and fills the pore of the spaces and cavities of the specimens. As I said, bones, shell, wood. So the pores of the organisms, tissues are filled with these materials that these minerals precipitate out of the water, and could be two types. That is not very kind of outstanding, this part, that precipitation when the minerals that are filling the pores, it's silica, or pyritization that involves the deposition of iron and sulfur. So this part is the first process of the petrification, the permineralization. And then we have the replacement. So the replacement is basically that the water containing minerals as well dissolves the original solid material of the organism and is replaced by minerals. So when there is a slower rate of this dissolution happening, means that there's better details of the structure that can be analyzed as well. And other kind of preservation that I mentioned at the beginning is an altered state, that the plant part is covered by fine-grained sediments, and this excludes the air and decaying organisms. What means that there is no compression, no petrification, but it's preserved very well with all the different characteristics of the cell as well. So what example do you think there is about that in plants? What is the hardest compound in plants? Hardest molecule. Sorry? Cellulose it's hard, but it's not so hard. There is another one that is. Sorry? Lignin is almost there, but there is something even harder, yeah. Nope. Close. So the hardest one is sporopollen. It's actually what is the walls of the pollen grains. That's why when you have very old fossils of angiosperms and you want to see like the origin and have inferences and hypotheses about that, all the fossil record is pollen. And that's one of the examples that could be conserved in an altered state. Would that be considered also an altered state? Yeah. Yeah, because there is no, yeah, so it's surrounded by the still very well preserved, and you can extract even material from there. Yeah, and for example, when you have this kind of a third state, you cannot only score morphological from the outside of the material, like in this seed that you can see here, but you can also do, for example, CT scans to see internal structure. This is the same seed that is there. As you can see here, we were interested in the shape of the embryo inside, and we can see some of the shape, how was the embryo core in that part of the seed. So in that way, some fossil with volume could give us much more information if there is a good preservation. Yeah. So other concepts are quite important in the way to study fossils is the fossil recovery rate. Because we were seeing different kind of preservations, and that's relating to the preservation rate. And it's quite different that to the fossil recovery rate. Like this preservation rate is related to taphonomy, and is a rate at which the lineage is fossilized. But then we have the fossil recovery rate, that is this rate at which fossils survive, are found, are collected, imaged, and also curated. So this also includes like how the collections keep, the management of publications, the funds available to find those fossils. That's more related to our economy sometimes, and politics, and science goals to find more fossils in different areas of the world. So, for example, okay. For example, in this is from the paleobiology database, and you can see like this is for all the fossils of plants, and this is the concentration of the plant fossils all around the world, and how most of the collections are clustered here in North America and Europe. So that's mostly because the major, like the larger amount of funds, explorations to find fossils, and how there is more ability of resources to be curated and keep in different museums. Other of the concepts that is interesting, is important to know, and this part is also, you can see it from a previous workshop. Thank you to Mike May and Carl Rofflet. I organized this virtual workshop that you can check online. And so in this way, you can have like different concepts of pool of the recent and pool of the present. So in a lot of molecular papers, you will see this term, the pool of the present. It's actually this constant or accelerating increase in the numbers of lineages towards the present. That's important, but then we have the pool of the reasons about the fossil record. That is mostly about, we have a better sampling in the modern than the recent past, mostly because the preservation is better as well. Taxa from the recent fossil record are likely to be extant well. And the rock record for the recent is much better. So there are higher discovery rates. We have much more fossils from recent strata than from very old times. So now we're going to move on to the geological timescale and what is important for our studies, for inference, for the inference of divergence times. Because sometimes, like, we have the fossil, we have the dates, and okay, let's do analysis, but it's very important to keep in mind each of these steps, analyze your fossils, the dates, as well as the morphological characters. So the geological timescale. Why is this important for divergence time estimation? Well, the fossils provide the dates. right, for our analysis. And these fossils come from rocks, and actually the change of the age of the rocks changes over time. And you will say, well, I thought it was a fixed date. No, all the methodologies are changing, are advancing, and we have better and better dates through the time. So for example, we have a little section of the chronostratigraphic chart here. And you can see how the different periods and how they are fixed with different ages. So the geologists and the paleontologists use these periods and stages rather than the absolute time. Because the period, like for example, if we say our fossil is from the Tortonian in the Miocene, it's going to be fixed, but the ages of that are updated all the time. So the periods and stages are based in the biostratigraphy, also radiometric dates, and the correlations between rock layers. And as I said, all these methods evolve over time and the dates change. So as a reminder, the name changes in the rock record also for local stratigraphic units, and as I said, for international chronostratigraphy. So it's important to keep you updated. And how do we do that? Well, first, using this web page where we can access to the most chronostratigraphic chart. That is this one, for example. So you can see here, this is February 2022. And usually all the dates are closer to now. The most recent dates are quite fixed and quite stable, but dates are changing more frequently are the ones that are more into the past. So let's see some interesting symbols and parts of this chart. First, we have this gold bike here. That is actually the global boundary stratotype section and points, and defines the lower boundaries of the unit. So it's defining where starts this period. Also, the numerical ages can change and do not define the units. And the boundaries with constrained numerical ages are indicated like the oldest one with this symbol. Sometimes we can see also this, that it has the uncertainty associated with the age. And that usually happens when it's older into the past. So for example, we have these fossils. This is one that we are describing actually from the tomato family, and it's a calyx fossil from the Green River Formation. Luckily for us, the Green River formation is very well studied. There is a lot of work, there is a good datation of it. And for example, in this work in this work from Utah, actually, we can see how the Green River Formation falls into the Eocene. But we always have to check most recent works with radioisometric dating techniques, for example, that can give us very precise dates for our straight our fossil was found. So it's a good practice to check the international chronostratigraphic date, but also check the regional chronostratigraphic, and also the establishing the regional lithostratigraphy. And as I said, always checking the newest dating techniques that have been applied to this rock. So something else about this nature of the fossil record and some terminology to remind is the difference between stratigraphic uncertainty and stratigraphic range. This uncertainty is what we usually use when we describe fossil, for example, That is when it's inexactness or uncertainty in the age estimate for a fossil or a fossil locality. And then we have the stratigraphic range. That is the geological or temporal range across which a lineage is sampled. For example, we can see that several fossils belong to the same lineage, and that will mean that we have a stratigraphic range with one fossil for one. Sometimes can happen, and sometimes we can have several. And this stratigraphic range spans from its first appearance datum to the last appearance datum. And remember that always that is underestimated, because having those fossils that give us the range for the appearance of this lineage doesn't mean that there are other fossils out there that could expand that range. So, and the problem with these two terms is that actually the dates, for example, are presented in the same way, usually. So it's pretty easy to confuse this terminology. Usually the dates are expressed like this, with a range, or it could be with uncertainty, but it could be expressed up. So we have to be very careful of what we are expressing when we include these variations. So a stratigraphic range, for example, is a good way to express it. For example, the group of ferns, since the first that appear to the last, because they are still extant, we can express it in this way. And that will be a really nice way to see your taxa, and also trying to establish how is that range for each of your lineages. And how do you know what fossils? Because you can have multiple fossils. They could have been described several fossils for each species, but sure that they correspond to the lineage where they were. Good way is doing some cluster analysis that we are going to see in a bit, to see if these fossils cluster to correspond to the same lineage. Then we can do a graphic such as a reminder and start working with our data like this to see, for example, if a lineage of tomato family, for example, Physalis, for where it started with the first fossils to the recent ancestors or to the current taxa. So these ranges could be helpful in estimating divergence times. This could include it in red base as well. We're not going to cover this during this workshop because it will be very long for today. But there's also tutorials in the web page of red base that you can see, you can check to do this. Remember always that this first appearance datum and the last one underestimate the stratigraphic range, as I was saying, and you can incorporate this into red base. So what is next? Well, we have the well-preserved fossils, we have the dates, the geological timescale. So the next is the morphology. So before that, if you have questions.